A pathovar flaccumfaciens of gram-positive bacterium Curtobacterium flaccumfaciens, or CFF for short, is the causal agent of bacterial wilt of soybean and other leguminosa plants, a disease that damaged many economically important crops around the globe. The photographs in this slide show soybean plants infected with the CFF. Previous genomic studies demonstrated the differences between pathogenic Curtobacterium flaccumfaciens pathovar flaccumfaciens and non-pathogenic Curtobacterium flaccumfaciens isolates, and this difference is probably related to the genes encoded pectate lyase, serine proteases, and cellulases. The main goal of the present research was to clarify the set and possible origin of genes responsible for CFF virulence. Our work has also addressed the problem taxonomic and evolutionary relationships between new species assigned to genus Curtobacterium using the latest genomic data. What is Curtobacterium flaccumfaciens? Is the first question of our study. The published taxonomic study of the genus Curtobacterium by Collins and Jones in 1983 was based on microbiological methods, and there is no work that included analysis of genomes for the related bacteria sequenced in recent years. Many Curtobacterium strains are classified by partial sequences of one gene. As the first step, we conducted the 16S RNA phylogenetic analysis as an essential step for bacterial taxonomy. We collected all available Curtobacterium 16S RNA sequences together with representatives for recognized genera of Microbacteriaceae family. By the end of May 2021, there were about 170 draft and complete Curtobacterium genomes in the GenBank database. The phylogenetic tree places almost all the Curtobacterium genomes in one monophyletic clade. The phylogeny of 16S RNA does not provide species differentiation of Curtobacterium strains, but distinguishes them from all other related genera. The tree based on 16S RNA sequences suggests that some Curtobacterium strains could be misclassified. Two strains under the name Curtobacterium were found to group distantly. They are Curtobacterium S6S and a strain assigned to so-called Curtobacterium fungi, the suggested name for wheat pathogen, not approved legally. The latter was represented only by the 16S RNA sequence. 23S RNA is longer and in case of Curtobacterium is less conservative. The 23S RNA phylogenetic analysis better resolves the distinction of different species. As well as 16S RNA phylogeny, the 23S analysis demonstrated the cases of misclassification. The 23S RNA phylogenetic trees were not able to place all the analyzed strains in distinct clades, but they grouped the known Curtobacterium flaccumfaciens strains in a single monophyletic group. The Ad hoc Committee on Reconciliation of Approaches to Bacterial Systematics in 1987 stated that there was general agreement that the complete DNA sequences would be the reference standard to define phylogeny, and the phylogeny should define taxonomy. Using whole genomes decreases the subjective errors related to the selection of the genes for analysis. The average nucleotide identity, N values, are commonly used in species delineation. Generally accepted threshold for the species identity is 95–96% of N. Neighbor joining clusterization but any distance grouped all classified Curtobacterium flaccumfaciens strains and a part of unclassified Curtobacterium in one large cluster consisting of 51 strains. The lowest N value in this cluster compared to the Curtobacterium flaccumfaciens type strain was 93.5%, which is lower than the species threshold. This large cluster included two subclusters. First of them included 43 strains with the lowest N value about 94.5%, and the second subcluster included 8 strains more distant from the Curtobacterium flaccumfaciens type strain and closely related with each other. Thus, the average nucleotide identity calculations demonstrated complex relations within the Curtobacterium flaccumfaciens cluster. Based on the ANI data, we should possibly divide the Curtobacterium flaccumfaciens group of strains into at least 2 close species. Nevertheless, the consistency of whole genome analysis can be hampered by the events of horizontal transfer. Such events occur often in the evolutionary history of prokaryotes. Therefore, the modern taxonomic studies apply phylogenetic analysis based on the concatenated sequences of core conservative genes. The set of genes used in this study included the ribosomal protein genes, amino acid synthesis, and other genes of translational and transcriptional cellular apparatus. These genes are less vulnerable to the lateral transfer. Indeed, the phylogenetic tree obtained with 91 core gene sequences easily distinguishes the clade containing Curtobacterium flaccumfaciens species. This tree basically corresponds to the any data. It places 51 Curtobacterium strains in a distinct clade. This clade includes two inner clades, consisting of 43 and 8 strains each. As well as the previous methods, this tree places pathogenic and not pathogenic strains together. Pangenome study revealed 570 core proteins with a similarity of over 50% presented in all the Curtobacterium strains. We constructed a phylogenetic tree using the concatenated amino acid sequences of those proteins. As well as 91 core genes phylogeny, this tree allowed to distinguish the flaccumfaciens species. The tree also matches the N matrix well. However, this tree demonstrated different topology compared to the previous one. It could be the consequences of different evolutionary history of different genes and gene clusters. As well as the previous tree, this tree places pathogenic and not pathogenic strains together, testifying of the reasons of phytopathogenicity probably not related with the core genome. Finally, the phylogenetic analysis and the whole genome comparisons allowed to distinguish 51 Curtobacterium strains presented in GenBank as related to Curtobacterium flaccumfaciens. In actuality, this group may be divided into at least two close species. The performed analysis highlight the need for taxonomy revisions. Using the published GenBank sequences, we checked the diagnostic primers recommended for the detection of Curtobacterium flaccumfaciens pathovirus. The test results showed that the primers were not suitable for the detection of all classified Curtobacterium flaccumfaciens species. We also checked the genus-specific primers constructed earlier for the detection of bacteria belonging to the genus of Curtobacterium, and found that they can give both false negative and false positive results. So we constructed a new diagnostic set for the conventional and real-time PCR for the Curtobacterium diagnostics. Besides, we developed a specific set of primers with a longer amplicon for taxonomic purposes. The first experimental results demonstrated that the developed primers worked with the sample collected well. The virulence of bacterial plant pathogen is often related with the so-called virulence factors, including such proteins as pectate lyases, cellulases, hydrolases, proteases, and other enzymes. To find the gene of virulence factors, we used the same approach we used earlier for search for taxon unique sequences. The genome of CFF type strain was turned into short DNA sequences, and the set of those short DNA sequences was searched against the database constructed with not Curtobacterium, not phytopathogenic Microbacteriaceae genomes. The pseudochromosome was constructed by concatenation of the assembled genome context. The sequences absent in not Curtobacterium genome were mapped on the pseudochromosome. The map of the pseudochromosome with those dissimilar regions is shown in this slide. The specific for the pathovirus flaccumfaciens of Curtobacterium flaccumfaciens sequences point to the genome region containing the genes which can be responsible for the CFF virulence. Many of those genes have close homologs in the genomes of phytopathogens related to Curtobacterium distantly, for example Clavibacter, Dicea, Pictobacterium, Xanthomonas pathovirus. Most of the supposed virulence genes were absent in not pathogenic Curtobacterium strains. The presence of transposases, plasmid-like and bacteriophage-like genes testify that the revealed region belonged to the pathogenicity island. Pathogenicity islands encode various virulence factors, and they are normally absent from non-pathogenic strains of the same or closely related species. The pathogenicity islands are defined as a subclass of genomic islands. The pathogenicity islands can be found in the chromosomes of a large number of bacterial pathogens. They are acquired by horizontal transfer via transduction, conjugation, and transformation, and provide quantum leaps in microbial evolution. The search for virulence genes in other pathovars of Curtobacterium flaccumfaciens found very similar pathogenicity islands in the genomes of Curtobacterium plasmids. Besides more distant but still similar mobile elements were found in pathovar of Clavibacter michiganensis. The latter is an actinomycete also belonging to the Microbacteriaceae family. We found 3 Curtobacterium plasmids containing the pathogenicity islands. Similar genomic islands were found in other pathovars belonging to the Microbacteriaceae family. The homologs of Curtobacterium virulence genes found in chromosomes of some other bacteria seem also to be acquired with participation of mobile elements. The genetic maps shown on this slide demonstrate the presence of plasmid and bacteriophage genes in the region surrounding the virulence genes. The virulence genes are colored red and the plasmid or bacteriophage genes are colored blue. The genomics of the Curtobacterium plasmids containing the pathogenicity islands is a matter of special interest. We annotated one of the plasmids, PCFF113, using both BLAST and HMM searches. The plasmid genome encodes typical plasmid genes and modules. The PCFF113 genome includes the conjugation gene module consisting of several genes, toxin-antitoxin system genes, metabolism genes, transcription genes comprising several RNase polymerase sigma factors, and an advanced replication apparatus. Also, at least 2 genes encode the antibiotics-resistant proteins. The replication apparatus of PCFF113 is interesting for the presence of DNA polymerase I, which has no homologs among other organisms, but some actinomycetes. However, the HMM motif comparison and the homology modeling demonstrated a high level of structural similarity of this protein to the DNA polymerases of the smallest known tailed bacteriophage Phi29, tectivirus, PRD1, eukaryotic adenoviruses, and to an unusual archaeal Acidianus bottle-shaped virus. All listed above organisms employ the protein primers in replication. Interestingly, the DNA polymerases encoded on the Escherichia coli and Thermococcus codecarensis chromosomes are structurally less similar to the PCFF113 DNA polymerase than in the cases of organisms listed above. The evolutionary story of plasmid proteins is very interesting from the point of view of theories assuming the common evolutionary origin of plasmids and viruses. The evolutionary history of pictate lyase, which is one of important virulence factors, was studied with phylogenetic analysis. We searched for the Curtobacterium pictate lyase homologs and constructed a phylogenetic tree with representative of different groups of organisms. Intriguingly, the tree testifies of multiple horizontal transfer of the pictate lyase both within actinomycetes and between actinomycetes and proteobacteria. It seems that the Curtobacterium genes were acquired through a chain of lateral transfers involving evolutionary distant groups of bacteria. The closest homologs of Curtobacterium pictate lyase were found in pathovars of Clavibacter. The tree also indicates the ancient events of horizontal transfer between bacterial organisms and eukaryotes, including oomycetes, fungi, and nematodes. In conclusion, I want to summarize the results of this work. To clarify the relations and to check the taxonomic assignment within the genus Curtobacterium, we performed complex studies comprising the intergenomic comparisons and phylogenetic analysis, employing different sets of genes. The studies allowed to suppose new taxonomic assignments for a number of Curtobacterium strains. Analysis of earlier developed PCR diagnostic kits indicated the need in new methods. A novel set of genus-specific primers was constructed, and first experiments demonstrated the suitability of the set for Curtobacterium detection. Analysis of the chromosomal and plasmid genomes of pathogenic and non-pathogenic Curtobacterium strains has identified the pathogenicity islands in the examined bacterial chromosomes and plasmids. The phylogenetic and genomic studies suggest an important role of mobile elements and the horizontal transfer and acquisition of virulence factors by bacterial plant pathogens. The presence of antibiotics resistance genes in mobile elements can testify of an efficiency of antibiotics treatment of the diseases caused by Curtobacterium bacteria. Techniques such as phage therapy can be tried to effectively treat these diseases. Thank you for your attention.